Why is menstrual blood still treated like a waste in modern medicine? Can we develop contraceptions that doesn't rely on hormones or surgery? And what if we could detect gynecological diseases earlier, at home, with just a simple PET? Well, these are not hypothetical questions, these are real scientific challenges that today's guest is tackling head-on.
So there is this hypothesis that menstruation blood actually flows back through the fallopian tubes and then, causes these lesions in some of the patients. And I was like, wait, this is very interesting. Maybe we can just use that implant. And he was like, oh yeah, why not? And I was like super pumped. And I was like, okay, this is a cool part of this project.
For decades, women's health has been sidelined in medical research. We still lack affordable, accessible diagnostics for conditions like endometriosis.
I think there's still a lot of stigma attached to it, actually shockingly much, I have to say.
Contraceptive options often come with significant side effects. And menstrual blood? Well, it's been largely ignored by the scientific community until now. Today's episode is about changing that narrative. My guest is Professor Dr. Inge Herrmann, a trailblazer working at the cutting edge of material science and biomedical engineering. She and her team are developing technologies that not only work in the lab, but that could transform real lives. From in-pad diagnostics that turn menstruation into a source of health data to smart hydrogels that could offer safer, reversible contraception, her research is pushing the boundaries of what's possible in women's health. So let's get into it. This is an episode you don't want to miss, so have fun and enjoy our conversation.
Daily dose of honest science.
Hello guys, and welcome back to Daily Dose of Honest Science, your go-to podcast for evidence-based conversation and biomedical science. I'm your host, Dr. Chani Kumar, and today my guest is Professor Inge Herrmann, a leading expert at the intersection of material science, biomedical engineering, and translational medicine. She is the founder and head of the Ingenuity Lab at the University Hospital Balgrist and the University of Zurich, and serves as associate faculty at the ETH AI Center, where she contributes to the advancements of AI-driven materials, innovation, and equitable technology development. Professor Herrmann is a professor at the University of Zurich, Balgrist University Hospital, Empa, and an accredited professor at the Department of Mechanical and Process Engineering at ETH Zurich. Her research focuses on development of clinical translatable technologies, including nanoparticle-based diagnostics and therapeutic systems, some of which we already have progressed to commercialization. She is the recipient of numerous prestigious awards, including the ETH Lattice Award and Science Breakthrough of the Year by Falling Walls and the American Chemical Society Rising Star Award. Her work has also been recognized with multiple innovations prizes from ETH Zurich and Empa, as well as awards honoring her leadership in equitable technology development. With an academic background that spans ETH Zurich, Imperial College London, and University of Illinois at Chicago, she brings a distinguished record of scientific leadership and interdisciplinary innovations. A big, warm welcome and applause for Professor Inge Herrmann. Thank you so much for joining today and having you as a guest on my podcast. I'm really honored and grateful to have you.
Thank you so much for the warm welcome and a kind introduction. I'm very happy to be here with you today.
So we start right away because we would love to get to know a bit more. So I would like to know, what was the key moment in your life where you said, I want to go into research, and specifically, what shaped your career path that you are now there where you are?
I think the explanation is quite simple. I think it was very much driven by curiosity and basically by wanting to understand things in great depth. And then also I like the freedom, and I like the diversity and the variability of research. Basically, you can work on whatever you deem important. You can find your problems that you want to work on, and you can find your collaborators you want to work with. And this basically allows creation of an environment and an ecosystem to your liking, or to our liking. And I think this is the most fascinating part about academic research.
Yeah, that's true. Yeah, I totally agree. And you're in the intersection between engineering, mathematics, but also translational medicine. So I would like to know why engineering. So usually people choose only biology or maybe molecular biology or biochemistry, but you were like, I want to create materials that do have a great impact inside the body, that they can help to heal wounds and so on. So why engineering is so fascinating to you, and maybe what's also a bit of the problems that you face, because it's not easy to engineer something that would work in the body, right?
Engineering is basically problem solving, and I like the engineering mindset because it's very solution oriented and very like goal focused. So basically, an engineer wants to solve a problem, and solving that problem is the highest priority. And so basically, all the steps build towards a solution of this specific problem. And I think an engineering mindset really helps a lot in staying focused, and first of all, understanding the problem like deeply, but then also defining the problem and defining design requirements for technologies, and then actually starting to build these technologies. And I think it's just a very powerful way of developing potential new solutions in a bit of a more structured way. And I think bringing these mindsets together, like the mindsets of a scientist, of an engineer, and a clinician, is not only powerful, but it's essential to actually build medical technologies for the future. Because in the end, I think people need to be supportive of these technologies, and they need to be involved in the design and the making of the technologies. And I see the engineering part as a sort of unifying feature. It sort of bridges the pure sciences with the clinics.
Yeah, that's really fascinating, and also what you have already published is what we will go to later, is like an example for that. Maybe could you explain to the audience, because I have a very wide variety of people, so some in the academics, some in industry, but also normal general public, what your current role is and how your let's say day of a life looks like in your daily life.
Yeah, so my current role, I think, is kind of broad, but I mean, I'm a professor, so I'm a teacher, I'm a researcher, I'm also a bit of an entrepreneur, and to a large extent, I'm also a mentor and a leader, manager. And I think all these components, they're like almost equally important, and their synergies are vast. And so basically, I like all of them actually, and I like the interplay particularly. And I think it's important to like understand all these roles, because basically, I mean, I live from teaching, right? To some extent, I live from teaching because these are like, we're educating the people that basically maybe in future work in our lab, or in future work in other labs or in industry. Yeah. So this is basically the foundation. And then, of course, the research is the core business, right? So we, like, a large amount of our time is dedicated to research, and it's really the core business of our lab. And this is where, the foundation is built of the technologies. This is where innovation happens. And then once this is done, like in order to really push the technologies and make them translate, this is where entrepreneurship or also collaboration with industry come in, because for medical technologies, the translation to the clinics is no easy business. And so it's best pushed by the people that invented technology and are very attached to the technology and care a lot about the technology. And so I see entrepreneurship as a really important part in making these technologies reach the real world. And so the entrepreneurship part is pretty much where my lab, ends, and then the new stuff begins. And then, I think all the rest is basically mentorship, leadership, management. This basically means it's just my job to make sure that these operations run smoothly.
Mm-hmm. And you said that entrepreneurship is also kind of part of your job. I would like to know if you had to do extra courses to get into the world, because we scientists don't really get into all these kind of topics, right? So we are more taught on the scientific side. So did you had to take extra courses, or is it something that naturally falls into you, that you can do it by just reading some books?
Yeah, I think it's pretty much intrinsic to my research focus, because basically we start with a problem, we start with a need. And so basically a need very often is also a business opportunity. And so we actually don't need to do that much to make sure, that our technologies find a market, because it's already implemented in the design process. And then, I learn by exposure, right? So in Zurich especially, we have a very lively, startup ecosystem, and, it's hard to not be part of it. It's basically impossible to not be part of it. And then, it's also our network grows through my involvement, but also through involvement of our students and of our postdocs. And, I think entrepreneurship is something that's not really taught well, or that you don't really learn in a course. It's very hard to learn in a course. And so you learn by experience.
And when you have developed something in the lab and you go into the business side, how much of this original ideas are shared among maybe PhD and postdocs and you? Because at some point you have to give away the rights, or do you have a patent that is with your name attached? So how does it function? Can you maybe tell the process?
So we typically have patents, but patents are owned by our employers, and so we are the inventors, but the employers own the patent, and so these patents are then licensed to the startup companies. And typically my involvement is operational involvement is close to zero, right? And it's really the former PhD students or postdocs that then lead the business. It's also I don't have time, and it's also not my job to lead the business, and it's also it's also a potential conflict of interest. So I'm out in that part, but of course I very closely work with them, and I'm typically their closest advisor. And so this is, it's pretty, straightforward how this can be done. And typically these people are also extremely motivated to push that endeavor.
That's really cool, because I was thinking that you maybe get some investment return or something like that, or somehow named at some point. But if you are quite out and the postdocs do the business, or the PhDs later on, then of course, it makes sense to give it away at some point. Your current role spans all these different disciplines and different topics that you have to overcome. The main question that I have is, what is your future goal? What do you want to achieve maybe at the end? I don't know, if you're 80s and you'll be like, I've achieved that. So what would be the ultimate goal for you?
So I think the ultimate goal is to maximize impact of technology, positive impact of technology. And so I mean, we're translating things, and I think we see impact of that translation, and like the goal is to basically repeat that, and really find a flow where we can repeatedly create and innovate like cutting edge technology that benefits like society at large, and that we really manage to identify the most impactful key areas, the areas that have the most pressing needs, and then make sure that our technologies really fulfill these needs and also translate. And I think statistically, this usually happens like once in a career. Typically, people manage to translate one thing in their career at best, I guess. And I'm really interested in, making this more repeatable by really defining and understanding these needs and the tech requirements and the regulatory requirements very deeply to, make that process more efficient.
And the needs are usually born from basic research that you identify in papers that you see, that could be cool if we could have like a solution for that. Or how do you get to know which need has to be fulfilled with a new technology? How do you get to that process?
I mean, it's mostly talking to clinical partners. I've never found a need in a research paper. So it's mostly talking to clinical partners and really like mostly also like friends of mine that are, like very exceptional clinicians that have a bit of an understanding of technology and like are open-minded. And so understanding like their problems deeply is a good source, I think. And then, sometimes they don't even know that the problems they have are problems, or they don't even know that there's maybe potentially even easy solution to the problems they have. And that's really our gap to fill. It's like understanding what are their pain points, and then also how we can match technology with these needs, and how we can also, in most cases, it's also how we can make sure that this can become a sort of profitable business, right? Because there's no translation without this being economically viable.
Yes, exactly. Yeah. I also do love the intersection with clinical people. It's just you realize how important science gets, and maybe realize also how important it is to do now translational research, which is now coming up more and more. So you are definitely a forefront figure in that. So I'm really astonished also by what you have built up till now. It really is something that is inspiring me to continue. Maybe off topic a bit, but besides your general interest that you have now in your research, what are other interests that you might have besides research? So what do you do maybe in your spare time?
Yeah, I think it's kind of, similar things. I mean, I love to explore. I love to travel. I love to cook. I love handicraft. I love, my little workshop where I can build things. I love my dog. My friends. I think also my friends are a really great inspiration for many of the things we do. Some of them work in very different fields. Some of them work in economics. Some of them are chefs. Some of them are opera singers. I think interacting with them is it's a great source of inspiration, and also a bit of a change of scenery sometimes. I think these are I think the mind sometimes needs a bit of a break. So I think constant focus is not always the most efficient thing. And I think especially for creativity, having different perspectives and different inputs is great. And I think there's a big element of serendipity in these kind of things. So Yeah.
Definitely. Yes. Yeah. And of course, you have also a family, and I would love to know from your side, how difficult is it sometimes, or maybe easy, it depends how you look at it, to combine your scientific career with your personal life. So sometimes, what is maybe sometimes very challenging for you, and do you find easy?
Yeah. So, I mean, for me, it's easy because I don't have kids, but by choice, because I love my freedom. So that makes it a lot easier. That really makes it a lot easier. So I don't get the sleep deprivation that some of my colleagues get, and I mean, I wouldn't survive it. Like I wouldn't have a brain if I wouldn't
Mhm.
sleep my 8 hours, I think, or 7, 8 hours. So that's yeah. I think this makes it really a lot easier. Yeah, of course, I have a partner, and this also sets to some extent some boundaries. But it's like it's so much easier. And I'm personally, I'm very happy with this choice, but I know it's not the choice for everyone. And I understand that
Yeah.
it can be extremely stressful. And I mean, I wouldn't want to imagine my life with 5 or 4 hours of sleep. So yeah,
Mhm.
all
Yeah,
the
true.
respect to my colleagues that are parents. Yeah, I think this is a whole other
Mhm.
challenge.
Mm-hmm. That's very interesting to me to know, because I can imagine that it would be much, much more difficult to get all your stuff done, and being all the figures that you're right now would be a bit more challenging. But it's really great to know how you manage that. So I would say we conclude this part already and get into your very exciting research. So thank you very much for giving the insights how you got into science, and sharing the passion that you have for it. So the second part is about rethinking materials for biological systems. And here I want to highlight the Inge Lab, which focuses on developing conceptual novel solutions for precision medicine through an interdisciplinary design thinking approach. And by combining materials engineering, advanced analytics, and simulations, this whole lab, or this institute, understands the interaction between engineering materials and biological system across scale. And this is a data-driven insight and enabling design of high-performance diagnostics and therapeutic technologies. And of course, it's a emphasis on real-world impact. And it's gathering all the experts from the University Hospital of Balgrist, University of Zurich, ETH Zurich, and Empa, and therefore it's really highlighting the collaboration between disciplines, and to solve complex biomedical challenges. And for this part, I chose two of your papers. And you're a big advocate for women's health, which I learned to know. And in your recent paper, which I find super interesting, was you developed a wearable in-pad diagnostic to detect biomarkers associated with diseases in menstrual blood. So first, the question what I had is, how did you come to do that? Like we all women, we kind of do this, and we never think of that we would test our menstrual blood for disease diagnostics. So can you tell a bit behind the scenes how you got into this, and wanted to develop something
Yes,
like
of
that?
course. I think it's, I had a PhD student, a doctoral student that came to my office, and he said like, oh yes, so in my PhD, I want to develop a blood biosensor. And I said like, okay, that's so boring. Like so many people are working on this, and I can't really offer you something that's, a unique angle. Let's maybe rethink that a little bit. And I said like, yeah, okay, maybe we can work on something else, like a biofluid is a bit more special. And, I said something like, oh, maybe we can work on feces or whatnot. And then he wasn't very pleased with that. And so I went home in the evening, and then the next day I traveled to work by train, and I sat in the train and I was like, okay, and I had my period. I was on my period, and then I looked out at the window, and I was like, wait, blood, period. Okay, maybe we could detect biomarkers in menstruation blood. And I was like, I took my phone and I checked what is out there, because I was like, okay, this is not such a, it's not such a non-obvious thing. And I was super surprised because no one had done it. And then I went to the office and I told him, and he was he was a bit hesitant, because he was like, okay, that's special. Like, also, like, he was a man, right? He had, I think, even though he's very open, it's not something that you talk about with your girlfriends. I mean, also I don't talk about it with my friends. So he was more like, okay, what is this even really, right? And then we did some research on, basically the literature, and we found that there's, even the process is not really well described. And so we decided to start working on that. Also because it's just, it's such an underserved area, and there's not even the basic things are known. So it's a very important area. And so this is how it happened.
Yeah, that's I would have imagined myself, that this was kind of the backstory. Maybe to know, it is quite hard because with menstrual blood, it's not just blood, right? It comes with tissue and everything. So what was like the most technical challenge that you had to develop something with this fluid?
I think the biggest challenge was the fluid control. Like, so the menstruation process is very heterogeneous. It's, as you said, it can be, it can contain tissue. I'm not going to say contaminate, but it can contain tissues. And also the viscosity of the blood can change over time, and the amount of blood can change over time, and with age and with other medical conditions. And so the process itself is very complex, and the amount of blood in quotation marks, the quality of blood can be very variable, and it's something you can't really control and you can't really predict. And it's also very poorly documented. And so, we built a lot of fluidic technology to actually handle that, and to develop an assay that's robust and that can, handle all these potential variations. And that was probably the hardest challenge, apart from, I guess the conceptual aspect that many people didn't think it was even possible. So
yeah, I could imagine. And what are now? So you have developed this. I think last year you published it. If I'm, yeah. So what are the next steps now? So I think it's already in the process of getting commercialized, I guess. So the question is, what kind of, let's say, biomarkers you can detect by now? Very reliable. What
so right now we have
this technique?
proof of principle for C-reactive protein, which is an inflammation and infection marker. That's, I think people know it from the blood analysis. It's not very specific, but it can still give hints, and whether there's an infection going on, or also potentially in Crohn's disease patients, it can have some relevance. Then we have a panel that's for cancer biomarkers. And again, we're not diagnosing, we're just detecting these proteins, and basically suggesting to the user to then go to the doctor's office for further tests. So the idea is that it's a sort of low threshold, affordable screening tool that can help you monitor your health. And so the biomarker panel is expandable. We're working on this with full power at the moment. I cannot reveal all biomarkers that we're targeting, but basically the cancer biomarkers and endometriosis biomarkers are definitely of interest. And it's a growing panel of analytes that we can detect.
And you already touched on the heterogeneity of menstruation blood. Did you had time to review what it would mean to have different ethnicities with menstruation blood? Do you see differences, or are there across different ethnic groups no changes or very
Yeah,
small?
I mean, it's a super interesting question. I wish I knew the answer. At the moment, we really have a bit of a bottleneck in the sense that, first of all, there's no other research really available that has looked into that. And I mean, this wouldn't even need, like, basically our part in this is we developed a platform, we developed the biosensor platform. But I mean, these questions, they're best answered by, by proteomics labs or by labs that really analyze the protein content. And so any kind of lab could do that if there is interest.
Giving me an idea. So maybe I follow up on this.
Really any lab could do that. Maybe people with better expertise even than us. And so, yeah, this is also something that we are looking forward to co-developing with the community, right? Because I mean, we can't run the show on all ends. And I think there are labs that are very well positioned to analyze menstruation. And there is a lab in Stanford that actually does that. And I think this is important work. That needs to be done. Absolutely.
Because I could imagine it has been shown that there are some disease markers different in ethnic groups. So either be Latin Americans or Africans and Caucasian people, that some markers that we think are associated with the disease might be different in other groups. So it would make totally sense to also check on that. Is there any comparable kind of diagnostic right now to yours, or are you really like forefront with these kind of things?
We are now not the only ones anymore addressing a menstruation blood, but we're the only ones that can detect things on the pad, like at home in one go, where we don't need an external lab. For us, this is an important aspect. This is a key aspect in our design process. I know other people try to build business in another way. We'll see who will make it.
And from the sensitivity and specificity point, would you say your product right now is the leading one, or do you would say there's another technology that is comparable with yours?
It depends. Like we're not a clinical chemistry lab, so the clinical chemistry lab will always be better and will always be the gold standard, and we're also not by any means challenging that. But what we can do is we can basically offer a test in the field. And since we're testing a fluid that no one else has tested, we're the only ones, so we are per definition probably the leading ones. This only like large field studies can really prove the validity of that approach.
And when people go to the doctor and give blood, usually you have it in coagulants like EDTA and so on, to detect biomarkers. How different is it for menstrual blood? Once it's given or once it's there, do we need something like this? Is like a layer of it on the pad, or do you think, or do you see that this is not necessary because menstrual blood itself is completely different? So what would you say to that?
Yeah, so far we don't think it's an issue. So like menstruation blood contains less coagulation factors, and we definitely benefit from that. Right now it doesn't seem to be an issue. But yeah, I wouldn't exclude it either, right?
And the biomarkers that you have now characterized to be fine for detection are still very stable in the menstrual blood. So you don't see like a degradation of it. Oh, that's really cool. How easy is it for the consumer to actually interpret the data? So that's maybe also a good question,
Yeah, I think it's
right?
super straightforward because we provide this app that basically helps the interpretation and it guides the interaction of the user with the technology. So yeah, I mean, this is also this is being designed in collaboration with the Institute of Design Research and the Zurich University of Arts. And so there's a whole team like looking at how this user tech interaction is best happening.
And is there a problem maybe with also like false positive results? Did you had something in that where you are looking now into it, or would you say it's quite straightforward and it's relatively low, the
Yeah,
false positives?
that's a very justified comment. Like it's a comment that we also get a lot. I think it really depends on the assay design and it depends on the specific biomarker.
Okay.
We think with a good assay design and a good biomarker selection, we think that this can be largely avoided. But yeah, of course, it's something to keep in mind. And yeah, we don't want to create more work for the healthcare workers. So there's definitely a point in this. But we're also very aware of that. So I think with this, yeah.
No, I could imagine I could imagine that for someone like me, if I would use the app, I know what to take care of. But someone who is maybe not so into science and understanding the readout, let's say, that she develops a certain anxiety, suddenly something is, I don't know, in the yellowish thing, and you should see your doctor, that people get a suit like an anxiety, or oh my god, I'm not okay, or something like this, and then it turns out to be not positive. So I think the app somehow is designed in such a way that people don't get that anxiety.
Definitely, and I think we have good precedents. So we have these skin checker apps that allow you to check your skin marks, and this is even more delicate, right? There is good experience with these.
Yeah.
So this makes me sort of confident that there is a good way of handling this if done correctly. And yeah, I mean, we still, I'm sure we'll have users that absolutely reject the technology. This is part of reality. And I mean, it's very clear that it's entirely in the hands of the user to either use it or not, and to what extent to use it. And also the data will not be shared, it will be local. And so I think this is really something that, it will self-regulate a little bit, in the sense that people that are interested in monitoring their health, they might, and others, they just simply choose not to. And that's also super fine.
Is there a possibility to join right now to test this, to give also some data from different people outside of Switzerland?
I mean, super soon. We get a lot of these requests. At the moment, we are still like finalizing the ethics license, so we can really, make sure that this field study happens in a best possible frame and environment. Because there are a lot of questions, right? So there might be accidental findings, and then so we need to know how to deal with these findings, and so there are a lot of questions that are very relevant also for the users and for the participants, and we want this to be a very positive experience for everyone, and so we're finalizing that at the moment.
Maybe from your personal point of view, why do you think is menstrual blood has been so neglected in the overall medicine field, or general in science? Why do you think it's such a underrepresented fluid to look at?
That's a good question, and like we asked ourselves as well. I think there's still a lot of stigma attached to it. Actually, shockingly much, I have to say. We got the funniest comments from our peers, anonymous comments, of course, but like comments ranging from, "Oh, this is disgusting. No one would ever do that. No one would ever use that," to like why do you develop this? It's not inclusive, and where's the equivalent for men? So we got really a lot of interesting comments, shocking comments, and I think it's just basically the acceptance of this biofluid is low in all parts of the world. In all parts of the world,
that's
and I
true.
think this is it's only revealed to me now that I see a bit how people respond to the research and how emotional their response is, and how everybody has an opinion, especially men and people that have never menstruated. Usually they have the strongest opinion, and they tell me exactly what is possible and what is not. And so that's interesting. I think it really reveals some of the structural issues we still have in our society that are maybe very hidden, but with MenstruAI we got to see quite a few of them.
So I can imagine. But also, did you get these comments on the research paper, like from the reviewers?
Some of them, like
really?
from the reviewers, we got two. Wow. One, this is disgusting, and the other one that menstruation blood is typically very contaminated. Like that, basically, it contains it contains feces.
Also
feces. They were like, it contains feces and urine, and most women are incontinent anyways. And we were like, pardon? So we got a lot of questionable comments that were not so supported by facts, and I think this is interesting.
I want to say thank you so much that you actually are doing this, because if you wouldn't be there, no one would do it. So I think everyone who is listening right now should thank you for going so forefront. And I could imagine, because there were some papers out right now looking at menstruation blood, because there have been some stem cells that are really important to look at. And now people are catching up and be like, oh, we could use these stem cells to reprogram certain things. Now menstruation blood becomes like the new go-to tissue that you could use to make all these stem cells. I don't think it's a bad thing to develop these. I really don't understand why the reviewer would write disgusting, because in the end, and I think every woman that is listening right now can totally relate on this, that how would you do a diagnostic without invasive, like having a needle inside, and draw blood is an invasive procedure. So this one is not very, in that sense, it's not invasive. It's actually, you just have a pad, in your underwear, and that's it. And I really don't get why people are like this. So we really, as you said, need a change in this whole discussion. So I hope that podcast helps a bit more. I think we could say that this is what you're doing is also like a broader movement towards women's health equality that you are right now doing. Maybe where do you see this platform being distributed? So do you think it's something that you would get from the gynecologist, or do you think it would be even in a way that you could buy it in a store? So where do you see it, maybe this kind of diagnostic tool?
Yeah, that's a good question. I think it's also something we're trying to figure out. I would want to have this as accessible as possible and as affordable as possible. I think at the moment I'm thinking about pharmacies mostly, where there's some kind of guidance, but not not so tightly regulated as if it, if you're at the gynecologist, you also can give blood easily. So I think it's probably step number one is more like a pharmacy, and then maybe step number two is more like a supermarket. I think this is more or less the distribution.
That would be, that would be really cool. You made another really cool invention, actually. It's a novel approach to tackle endometriosis and contraceptives, because you have, so usually let's say you revolutionized like contraception and women's health more broadly, because usually fallopian tube occlusion, which is essentially blocking the tubes that connect the ovaries to the uterus, is a common method of permanent birth control. It prevents the egg from the sperm from meeting and therefore stops fertilization. But if it didn't have to be permanent, so what you did now, a very cool thing is a material like a hydrogel that gently blocks the fallopian tubes without surgery, hormones, or long-term side effects. And I find this very interesting because so it was made for contraception, but then you realized that it was actually also interesting for endometriosis. So can you explain what is this gel made of and how it actually works?
So it's a polymer-based gel. So it contains macromolecules and mostly water, actually. And so it's a super soft gel. It has the consistency of a gummy bear. And initially, it basically has the size of a rice corn, and it basically has the rigidity and the size of a rice corn. And then once it's placed with a tiny endoscope, like hysteroscope, it absorbs the fluids from the fallopian tube, and then it swells. And so it swells a lot, 10 to 20 times, and it becomes super soft. And basically, by becoming large and super soft, it adapts the anatomy of the fallopian tube and then occludes the tube in a very, very gentle way. And the mechanical properties of the gel match the ones of the tissue. And so basically the tissue doesn't even really not realize that there is an implant. And so it's an acrylic polymer that's very stable, but it contains some crosslinks, so some connections that can be degraded by the application of light or the application of artificially introduced biological stimulus. And so the material can be degraded and liquefied on demand. So this is how it can be reversibly removed when contraception is no longer wanted.
And how was the design process of finding the perfect biomaterial? I could imagine that it was a long process to find something that really would match the tissue, it doesn't damage it, but also find a polymer that is, of course, not toxic and something that wouldn't disturb the overall anatomy of the ovaries, right?
Yes, I mean, we have a team of excellent chemists, polymer chemists, that were concerned with the formulation of this material. And then it really, we brought all the people to the table. We brought the gynecologist, we brought the medical device engineers, and we brought the chemists to the table. And then, we basically designed the implant to fulfill the properties we thought were important. And it turned out quite well. So we were very happy. We were very happy with the absence of an immune response, and we were very happy with the stability of the gel. But it was really all due to really good interaction between the chemistry team and the gynecology team.
And can you maybe say what was the time span of finding the perfect material to actually have the hydrogel? So what, 1 or 2 years, or was
I think 1 or 2 years sounds about right. I honestly don't know exactly, but I think 1 or 2 years sounds about right.
Well, that's I'm just fascinated by that. This is all possible by this gel. And you also tested it in animals, and has been shown that it's really not invasive, and also helps the helps or doesn't damage the tissue. And I think it's not in clinical trials right now. It's just you want to have it out there. But do you think it would be a good way to test also in broader human trials this hydrogel, how it works? Because of course not every ovary is the same. Would you say there's a
We don't know. I mean, we first need to show in chronic systemic toxicity studies that it's really safe. It's complicated and expensive, also because these are typically young, healthy women with a prospective lifetime of 60 plus years, or prospective additional lifetime of 60 plus years. And so basically, we need to somehow show that nothing bad happens, and this proof of absence is very hard. And so at the moment, like, we're just preparing to make the study design to hopefully show that such a technology can be safe before we can even think about human trials, because I mean yeah. So a new technology is, as much as I'm excited about it, a new technology always brings risks. And I think, it's different if the risk is, in a cancer patient that has like 1 more year to live without that treatment. It's a completely different story to like a 18-year-old woman that has a life expectancy of 85, and then we put an implant, and we just hope for the best. And I think this is really something that needs to be very well understood. I mean, it also needs to be understood for other contraceptives. Just small side note there. It's also not really fully understood, so there's a lot of work to do in this field. But I think it's just it's important to understand the potential risks in a best possible way.
And can you explain shortly why would you have a degradation by light? So how could we imagine that?
Yes.
Is it?
Yeah, it's simple. It's just bonds that break upon light application. And so this is basically a feature that we integrated into the material, so we can on demand break these bonds. Okay. And then basically the jelly, the gummy bear, becomes completely liquid and then gets excreted through the vagina.
Okay. And then it's out. And what do you think would be the lifetime of such a hydrogel? So like, is it, let's say you can keep it for 10 years, or do you need to renew it like every, I don't know, 5 to 5
yeah,
years?
That's a good question also. Like, we're targeting something like an IUD at the moment. So it's probably hopefully years, but it's not going to be 10 years, most likely. And it's also, there's something you can only find out in a real organism. And so in our case, this would probably be a very happy pig, but it takes time and it takes money.
What do you think is the most difficult, let's say, step in the commercialization, but also getting it to the patients, besides the clinical trials, of course? What could be something that is still very challenging for you right now?
Yeah, I think it's really, the single most challenging bit is really this safety of the implant, long-term safety. And it's very hard to answer. It's not, we have to extrapolate, and extrapolation is always risky. And so we have to extrapolate, from pigs to humans. and then we have to extrapolate from months, maybe years to decades, and it's something that's very hard. And
the polymers that you used, are they already used in some other cases where people already know the data, or is this something that have not been used in humans at all?
Yeah, not exactly this formulation, and this also makes it a little bit complicated. Yeah,
Okay.
but I mean, there are very few actually. There are very few formulations where we have good human data on, and so this makes the whole process very complicated.
Maybe so we talked about that the hydrogel helps in contraception, but it also helps in the case of endometriosis. So can you maybe explain why it got this dual role and how you got into that it could also help in endometriosis?
Yeah, that was another interesting chapter in our lab. So we had this polymer chemist and this gynecologist working together on this contraceptive technology, and it was kind of exciting. But then, I was like, yeah, okay, it's an obvious thing to do. Okay, let's do it. And then I had a conversation, actually during the break of my lecture, Lydia, I had a conversation with our gynecologist just because he was co-teaching with me. And then he was like, oh yeah, that's so funny. So there's this hypothesis that menstruation blood actually flows back through the fallopian tubes and then, like causes these lesions in some of the patients. And I was like, wait, this is very interesting. Maybe we can just use that implant. And he was like, oh yeah, why not? And I was like super pumped. And I was like, okay, this is a cool part of this project. This hypothesis is still, it's so-called Samson's theory, and it exists for 100 years, and it's still the prevailing hypothesis, but it's still a hypothesis. It's not really solidly proven. It's the one prevailing hypothesis though. And now, I mean, like at the bare minimum, using this technology, we can at least investigate the role of this retrograde menstruation. So in endometriosis. So there's a bare minimum we could achieve with this implant, if ever applied in humans. But I mean, we talked to many endometriosis experts, actually the key opinion leaders around the world, and we pitched the idea, and they were like, yeah, that sounds cool. It sounds like worth investigating. Whether this will like reduce the disease burden, we don't know. And it's also something we can only assess in human, and endometriosis is a human disease. And so we will only know in the very end whether this brings comfort and benefit. But we see that based on the requests that we get from many patients, we see that basically their suffering is enormous, and many of them write us messages saying that they're willing to sign any papers now. They don't care about any of the risks. They would just basically want to get the implant now. Okay. And I think this illustrates a bit how much they suffer and how few options are out there.
Yeah. Yes. Yeah. Usually, the question is how far before you need this hydrogel to actually prevent endometriosis, because I could imagine if you already have endometriosis, you might stop a bit more the inflammation from ongoing stuff, but something that has already been done or is ongoing as an inflammation, I don't think that it might stop that, right? So it's more like preventing more inflammation if someone already has endometriosis, or?
We still don't know it. I mean, there are so many hypotheses. I think it's what is kind of, I'm not going to say established, but close to established, is that these lesions can grow back. And so the question is now, what is the role of de novo formed lesions, and what is the role of lesions that grow back? Also, that could be investigated using the technology. So at least it could be disentangled, which is also interesting. But yeah, I mean, I think I don't even want to speculate too much, because I think it's just building on hypothesis. And I think we also saw in the past that some of these beliefs are also a bit problematic. And so I think the way we see it, or the way I see it, is that we really go back to, almost first principles. And we basically don't make any assumptions other than that we want to test the Samson's hypothesis. And I think this will be a little bit the starting point of that study.
Mm. And do you know what kind of endometriosis models do exist? Do you know something about this?
Yeah, we looked into it.
Like animal model?
Yeah, we looked into it. Very few of them are convincing, actually. None of them really. So there are mouse models where it transplant, but it's all artificially generated disease models. And then there's a monkey model, especially in Stanford. Yeah, but that's hard. I mean, it's very hard.
Okay.
And it's still, I mean, it's a human disease, and I think it's a little bit similar to, not pathology wise, but like inadequacy of disease model wise, it's a little bit similar to sepsis, where basically all the experimental models have so many shortcomings that things that work in these models translate very poorly. And then there is also a bit of an ethical question, like, how much meaning is it really, does it really give if you do animal trials and so on. So that's another challenging part. But I also know that pharma companies, they're also looking into that, right? They're looking into, what models are the most human-like models, disease-like models. We're observing.
So there's a huge
we're observing the field very closely, actually, to see what models might be good models.
But in the end, if it's something that is only human, maybe at some point I'm not sure if organoids would help at some point. If we could maybe make a organ on a chip or something like this, maybe that would be something in the future. Exactly. So maybe if you imagine the next 10 years, what do you hope, or what kind of impact do you want on women's health globally? So what would you like to achieve?
I think I'm like
with these two.
Yeah. So I'm very I would be very happy if actually at least one of these texts has translated successfully. I dare to predict that MenstruAI will be much faster and easier and lower risk. And I'm sort of hopeful that at least by illustrating that there is so much room for innovation, that this will get things moving a little bit more in that space, because we see a lot of we see pharma, like hormone-based approaches, like a lot, but we don't see much else. And I think there's a lot of room for technology, even simple technology that can change a lot for patients that don't really have options. And I think it's this is a primary goal of that research line. We want to actually change things for the patients.
Yeah, that's really like amazing what you're doing. And on top, maybe I would like to know from you, what are you currently else working? What are current projects that you would like to share with the people, where you're excited about?
Yeah, that's a good question. I mean, I'm always super excited. I don't do projects that I'm not excited about, but I mean, I'm super, I'm super excited about the project that we have on a surgical sealant that seals gastrointestinal leaks and has some chemical sensors integrated that basically alert the surgeons of potentially upcoming complications. That's now in translation. Very advanced translation, so it's part of the Zurich. And so I personally think that this has the potential to have, clinical impact, just because many of these patients also really suffer and they don't really have options. I'm very excited about some of the robotic surgery tools we're developing, like with sensors and like basically we're hoping to really also make like robotic surgery a lot smarter with like both the interplay of spectroscopy, but also materials to repair tissues. We have. Yeah, I'm not going to go into a lot of details, but yeah, we have other projects in women's health. We have a project that we think will help to cure bladder diseases, ranging from urinary tract infections to cancer to bladder pain syndrome. We have a project on postpartum hemorrhage. I think, we really have identified a number of very urgent unmet clinical needs, and we have partners in all of them. And we're working very hard to hopefully make these technologies useful and also translatable.
Yeah, that's super exciting to know. And I'm looking already forward for the next publications that you're trying to bring out, hopefully positive, not having disgusting comments on it. Maybe I would like to wrap up our conversation already, but of course, I would like to know from you what kind of challenges or opportunities do you have seen by doing this interdisciplinary approach now over so many years? What is like the takeaway message for the people? How difficult or maybe easy it can be to have these interdisciplinary people?
Yeah, I think I wouldn't say it's easy and I wouldn't say it's accepted. So definitely I got some very nasty comments, like including something like, oh, people that are not good at anything, they work in an interdisciplinary way or something like that. Yeah, it's okay.
Oh God.
I find it very funny actually.
Oh man.
But yeah, it's very common in the old school beliefs, and a lot of people, we're also at the very extreme end of the spectrum because we work in all kinds of diseases and with all kinds of materials and with all kinds of techs. And of course this, it's not, it bothers a lot of people. I think it's not an easy way. I also don't recommend it to anyone that, to anyone that takes comments very personal. I really don't. Typically I'm on a mission and I don't really listen to comments like that, but there are definitely easier ways. And I think one challenge with interdisciplinary research is also that you don't really easily have your community, because you're always a little bit the weird one, right? And I think unless you're very comfortable with that, which I am, I'm oddly comfortable with that, but I know that not everyone is. And I think, I think especially in Europe, interdisciplinarity is still in many institutions, it's not really lived very well. And it's not really very well accepted. I think I see a bit more progressive movement in the US. I don't know as long as we still have the US, but
exactly, yes.
I see that this interdisciplinarity is lived a bit more there, and I think it's for me it's the future, especially in fields like medical innovation. I think it's really essential that we bring all people to the table. And it's so fun. I think if you're curious and you love new things and you love to learn, I think it's the best thing that you can do.
Yeah. So we both share the same passion. I'm so happy to have you here. Maybe what could the community do better to improve the technology in women's health? What do you think, except, of course, doing collaborations, but what do you think we need to get the women's health a bit more into the center? What do you think? That we don't get these nasty comments.
Yeah, I think it's a good question. I think it's somehow like once you realize it's kind of obvious, it's kind of shocking that not much is done. I think basically it's important to also do fundamental research to really also understand disease mechanisms and also I think many of the issues are very hidden. Actually, one big thing is also like childbirth and everything around it. There are a lot of massive issues, ranging from the fact that the C-section is not a tiny thing. Like basically, society makes you believe that C-section is just a tiny scratch and you put on a Band-Aid and you're fine, which is not true. And then, also, there are so many extremely problematic things, including the husband stitch, and there's so many really, really scandalous issues. And I think we should talk about them. And I think for me, there's one thing, like women speaking up is one thing that's gonna change a lot, because honestly, the reason how I got to know about these clinical needs is because I hired Thomas, like a gynecologist, hired him in my group. And this is how we got to know about these problems. If he wasn't there to tell me these stories, I wouldn't even know about half of them. And I think this, being a woman myself, right? So I think this really shows that basically by being silent, and this is not by choice, right? I think this is really by societal structures. But being silent is a bit of a symptom here. And I think this is, what I really wish for, that women are not silent, and they don't feel like they need to hide all these things, because they're clearly unfair and they're clearly unacceptable. And I think by that, there's already by shedding light on this, there's already a lot of things that will change, just because it's unacceptable things happening.
And we are talking about it, making it publicly available. So I hope this podcast episode helps the people also to see the need, that we need to put the women's health more into the center. But we are coming now close to an end. And Professor Herrmann, I would like to give you some minutes to address something that you want to maybe discuss, or maybe a topic that is very dear to you. Feel free to talk about any misconception or misinformation that you have been seen in your scientific career. Would give you now the stage for it.
Thank you very much. Much appreciated. I think I already mentioned many of them, things that I feel very strongly about. I think what really is a great thing is to bring people together and, collaborate. I'm very open to discuss always with everyone. We love collaboration, and we love joining forces and building allies to, change the landscape together. I think this is really the most powerful way how we can lead progress and how we can change things, especially also for women in the healthcare space. And I would just like to say that we're open, and we're happy to discuss and support, both by collaboration, but also in any other way. We're always happy to provide visibility to women's health. We're also happy, of course, to talk to affected patients. I think all these diseases deserve more visibility, and there's nothing better than personal stories to illustrate the suffering and the personal impact that some of these things have. I think this is really, it's a bit of an open invitation to join forces.
Thank you very much for this. So maybe if you at some point want some patients to join, or some people who would like to test it, just let me know. I will happy to share your links that people can be inside. It's always good to have the variety of people. So we are actually at our end. Professor Herrmann, I want to thank you again for your time and this wonderful conversation that we had. I know you're very busy, so yeah. And if you got interested in her work, please make sure to follow her on LinkedIn, and links to her website and so on you can find in the description box. And of course, if you enjoyed our conversation, please make sure, if you like and want to see more of great episodes of future scientists and medical professionals, please make sure that you subscribe. Professor Herrmann, I have a slogan at the end of my podcast, and I would love to know if and feel honored if you say it with me together.
Super, of course.
Okay, good. Let's do it. 1, 2, 3. Let a little dose.
Second.
Okay. So it's let a little dose of honest science in your life and brighten up your day. And I always say it with my guests together. So if you would have time, it would be good. Okay, 1, 2, 3. Let a little dose of honest science in your life and brighten up your day. Yeah, exactly. Thank you very much. And we see us in the next episode. Have a great time. See you. Bye.
Thank you so much for listening.
Daily dose of honest science.